everyone. Today I will be presenting the preparation and optimization of a pediatric palatable praziquantel alcohol-free solution. And as many of you may know, praziquantel is a highly hydrophobic drug and currently is only available a solid formulation of 600 milligrams. This implies that often this tablet will be split for dose adjustment or even crushed and pulverized in order to be resuspended and administered to pediatric patients, which can lead to an inadequate dosing. Therefore, the purpose of this work was to develop an alcohol-free oral liquid formulation of praziquantel. First, we optimized the co-solvent mixture based on N-methyl-2-pyridone polyethylene glycol 400 and water via design of experiment approach. We applied a simple centroid mixture designed to study how the different proportion of these solvents can influence in the final concentration of praziquantel. Then we perform a palatability assay where we try different flavors and sugar concentration and combination in order to determine which can mask the bitter metallic taste of praziquantel. And finally, we perform an stability assay of the final formulation at different temperatures at 4, 25, and 40 Celsius degrees with and without light exposure. We study the stability in terms of concentration of praziquantel, pH value, and formation of any degradation products. Microbiological tests were also performed. We followed the guidelines of the United States Pharmacopeia for non-sterile products. The result here on the optimization of the solvent mixture. In this graph, we have the response surface. As you can see, when only N-methylpyrrolidone was used, we achieved a higher concentration of praziquantel solubilization alongside with polyethylene glycol 400. And as you can see, as the water proportion increased, the solubilization of praziquantel decreased. This can be explained by the highly hydrophobic nature of praziquantel. For the optimization process, following the mathematical model that we obtained from this design of experiment, we determined that the best proportion of co-solvent was this one that you can find on the screen, is 67% of N-methylpyrrolidone, 17% of polyethylene glycol, and 17% of water. With this proportion, we achieved a final concentration of 40 milligrams per milliliter of praziquantel. And as I said before, we also perform a palatability assay, and we find out that this combination of cherry flavor and sucralose were the best combination in order to mask the bitter metallic taste of praziquantel. We also add methylparaben sodium salt as a protective agent. Then, during the stability assay, as you can see in this graph, those samples that were stored at 40 and 25 Celsius degrees, we were able to keep the initial value, the value of the concentration of praziquantel. But on those samples that were stored at 4 Celsius degrees, you can see that we lost 5% of the initial concentration after 90 days, and after 4 months, that concentration decreased at 13.5%. The pH value of the final solution kept the initial value of 7, and also the microbiological tests that were all negative during this period of time, and we weren't able to find any product of degradation. So we can conclude that we developed a pediatric formulation reaching a final concentration of 40 milligrams per milliliter of praziquantel. Furthermore, bitter and metallic taste of praziquantel was masked, making it more tasteful for children, which could help with pediatric patients' compliance in different treatments such as intestinal diagnosis. Thank you very much for your attention.